I'm Toby Stovold from Scion. I'm presenting today on the status of specialty purpose breeding in the SWP program. In the Specialty Wood Products Research Partnership, we focus on three main areas: Douglas fir, eucalypts, nightens, regnans and fastigata. The cypresses, we do loose tannica, macrocarpa and hybrids. The SWP program also does durable eucalypt species, but that will be covered in a different presentation. Moving on to Douglas fir. Douglas fir breeding started in 1957 and 1959, mainly with Providence material from the Washington, Oregon areas. There were more trials in the 1970s, 1990s and in the 2010 era, including a major introduction of Californian provenances in the mid-1990s. Program has also established large plot genetic gain trials in 2012 to validate the gains from various seed orchards and generations of material. In 2018-2019, new trials were established which incorporated material previously being run in parallel by Earn's Law 1. More recently, genomic research has started using two trials. This involved collecting DNA from 2,200 trees, which were then genotyped on an axiom array, and this was in collaboration with Glenn Howell from Oregon State University. And we have 2,000 more samples awaiting analysis. So if you look on the right, the two trials were Kaingaroa and Gowan Hills. If you look at the chart in the slide there, the dark towers are the prediction accuracy of the breeding values when we just have the phenotype data, that's data from the tree, and the grey towers are when we add in the information that comes from the marker analysis. So in all cases, diameter, needle retention, stiffness, we got a better result by adding in marker data to the pedigree data. This year we'll use a very similar approach to estimate Swiss needlecast breeding values for most of the grafted parents that are in the various orchards around the place. Moving on to cypresses, I'll cover loose tannica, macrocarpa and the hybrid work that's happening. For loose tannica, breeding trials began in 1984. We had some failed second generation trials in 1998, which was a combination of goats and a not very fertile site. The second generation was repeated in 2006, and then recently in 2017, a third generation of tests was established. Along the way, a clonal program was initiated in 2002, as cypresses are amenable to propagation by cuttings. Currently, the clonal program is parked. We're not continuing on with that. From all generations of this research, the best material has been grafted to orchards, so good quality seed is available for those who wish to grow loose tannica. Macrocarpa, the first breeding trials were 1984 and a second generation in 1999. Similar to loose tannica, we started a clonal program in 2002, and also this program is currently parked. We're not investing in creating more clones. For this species, interest declined due to canker issues and planting dropped away. However, we noticed some tolerant trees in our trials. So in 2010 and 2011, we selected some parents that looked like they were tolerant in trials in Guavas, Longmire and Birch Hills, and these were grafted to the orchard of proseed. Over time, the seed orchard flowered and developed seed, open pollinated, and Pro Seed did some control pollinated seed. And in 2018, there was enough interest in SWP to get this material out. So we established 3 new progeny trials in 2019, Tarawera, Manawatu and Foxton. And we'll be monitoring these to see if they get canker infection, looking for tolerant families. Finding tolerant families will rebuild confidence in Macrocarpa and get people to plant it again. Cypress hybrids, the breeding plan we have calls for hybrid swarms, which is everything crossed with everything. Our main interest is in hybrids with Nucatensis, which would add durability. Nucatensis is class 2. Pollen was supplied from the BC breeding program in collaboration with John Russell. We also used Arizonica and Guadalupensis, thought to be tolerant of canker as well. And we crossed those to our best loose Tanica and Macrocarpa from our progeny trials and clonal programs, and also with them every combination we could get. Evaluation trials of this material were planted in 2009 and then again in 14, 15, and most recently in 2017. So far, we've evaluated 130 hybrids. 30 of those we identified early as being good in the nursery, they're good propagators. And from within those 30, we released 6 new Avenzii type and 6 new Leyland type to the market so that nurseries could grow store beds and have this material available for growers. Most recently in 2019, we added 2 sites of these new release material to look at matching species or clones to site. And that brings to a total of 8 sites we have with these 12 released hybrids. We have 150 additional hybrids yet to be evaluated. And we recently got some input from Farm Forest Association through their Cypress Development Group on what they thought would be ideal types for growing. Cypresses, what does the future look like? We need to identify canker-tolerant Macrocarpa that will rebuild confidence in growing the species. We'll maintain the loose Tanica breeding program, which is pushing on with growth form and monitoring canker. And most importantly is learning about the heartwood and durability of these new hybrids. The hybrids have a lot of potential, and testing to see how much durability we may have gained by bringing in Nucatensis. We need to investigate that and quantify it in the future. Moving on to the eucalypts, I'm calling them industrial eucalypts because they're not durable, and certainly our initial interest was for the fibre side of things. There will be some discussion about solid wood products from these species in another talk, and our species are Regnans, Fastigata and Nitens. So Regnans breeding started in 1978. The current breeding population size is around 100 families. The last round of tests we put in were 2009 and 2010. The population is currently sitting in 3rd generation. We've assessed these trials for growth and form, and following on from that, new material was grafted to orchards. As a backup to increase possible seed supply, if there was more interest in Regnans, we've established some gene resource plantations, and we plan to convert one of the trials into a seed stand, just in case the seed orchards can't meet existing demand. Fastigata, very similar story, started in 1979. This population's a bit bigger, it's 200 families. So we've used a rolling front strategy, which we split the population into 2 groups, which helps spread the resource requirements for testing. So the first half, which was 2 trials, has been assessed for growth, form and stiffness. And just recently we completed the second half, which was another 2 trials, and we'll be reporting on that later in the year. And as a result of that report, we expect to identify some new orchard candidates. Seed orchard seed, yep, it's available from the previous generation, and similar to Regnans, we will convert one site to a seed stand as backup in case there's more demand for the seed. Eucalyptus nitens, this is our one we spend most of our work on. The previous breeding population turned over in 2014. It's a rolling front strategy similar to Fastigata. We have about 220 families involved. We've done quite a lot of wood properties studies in one half of this population. We felled 780 trees. We looked at growth strain and shrinkage. And from each tree that was felled, we collected DNA for some genomic studies. We identified a bunch of new parents for seed orchard use, and further work will be to use the Scion spot to create NIR data and create some models for non-destructive sampling. For the genomics, 700 trees, again, SNP chip was used, and similar to Douglas fir, the accuracy of predictions was improved. If you look in the chart, the black towers, again, are just the pedigree or the phenotypes, and the grey towers are phenotype and the marker information together. So we had improvements in diameter, growth stress, shrinkage and density. Another thing with nitens is it's limited by Propsis. So with the cooperation of Southwood Export and Scion collected adult beetles from Poronui and Taupo. They were segregated into pairs and released into the forest. November 18, we had to wait, checking monthly that there had been damage worth assessing. And in May 2009, we did. We used a score from 1 to 10. 1 was no chewing, 10 was heaps. Turns out it's heritable at 0.15, very similar to diameter. There were some differences between orchards and other sources used, which means we've got the potential to select for browsing tolerance. The trial won't be sprayed this summer, so hopefully we can reassess it. If you look at the graph in the right-hand bottom corner, there are parents that are above average for growth and less browsed. Why does FGR and Scion invest in these? Well, 90% of the forest is radiata, so it's risk mitigation. Some sites are not suited to Douglas fir, the cooler sites, and some species are better for the chip market, and that's our industrial eucalypts. Specialty timbers, cypresses, that's a good alternative for radiata pine, and availability. There's good quality genetic seed from the program through the various orchards at ProSeed, Earnslaw One and Southwood Exports. And thank you very much.